let's learn this integrated human neural organoid atlas. So back to all these papers. So these were actually 25 publications. There were 36 data sets, so some publications had multiple data sets. 3 of them were actually unpublished. They were not yet a paper. And the data was curated to have consistent pre-processing. The metadata was harmonized, and then it was integrated also using auto-annotation. So we developed a tool to actually automatically annotate it and then also have this label-aware integration. And so, yeah, this was really teamwork. There was Jonas Fleck, who's now, he was a PhD student in my lab, now working with Greg Camp, Chisong, a postdoc or senior scientist in my lab, and Leander Donny in Fabian's lab, that were really kind of as a dream team working on this. And so now here you can see the data. Actually, this already was shown earlier today. So the organoid ages really span the 4 days, the very early, up to 300 days of organoid age. We had all kinds of different single-cell methods, 10x genomics, there were BD Rhapsody, homemade drop-seq, quad-seq, split-seq, smart-seq, so all kinds of different methods. The organoid protocols were either unguided, as I showed you in the beginning, you just let it differentiate, and there's actually can be line variation and so on. The organoid will establish somehow a patterning axis and develop different regions. The guided protocols where actually morphogens are added. Yeah, so it was this whole combination. So how does the atlas look now? A lot of plots here, but so this is the integrated neuroorganoid atlas, where now we can see actually starting from pluripotent stem cells, very few of them very early on, the neuroepithelium, then developing into stems, neuroprogenitor cells of the dorsal telencephalon, the ventral telencephalon, and all these non-telencephalic progenitors that then develop into their respective neurons. Here are the cortical excitatory neurons, the ventral inhibitory neurons, and the non-telencephalic neurons. If we zoom into this branch down here, there's actually quite some diversity. I mean, this is not just one cell type developing. There's actually all kinds of different ones here labeled just whether they're inhibitory or excitatory and what is one of the major marker genes. But actually, we will come to an annotation later when we actually map this to the primary atlas and try to have a annotation based on this shared embedding and label transfer. So the guided and unguided protocols are quite nicely mixed. It was just that unguided, this was our data set early on. This just existed only from the unguided, but yeah, generally they are mixed. Here you can see the ages. I should mention there are also astrocytes, there are oligodendrocyte precursor cells, so you have also the macroglial lineage present, and also mesenchymal cells, these kind of, they emerge in the organoids usually at a small proportion, also sometimes some neural crest cells that are maybe kind of off target or, yeah, depends how you see it. And then here's all kinds of marker genes on this embedding. If we now look at actually which organoid came from a guided or unguided protocol and what is the composition of that organoid, and so now here you see these stacked bar plots that show you the composition of these organoids. You can see that generally the unguided ones have a big mix of colors here, whereas if they are guided to the cortex, yeah, you mainly have these blue cortical cells present. And then here's guiding, for example, to mid-hindbrain, which, yeah, then you have more of these reddish colors, and so on. So overall, there's a trend. If you guided it towards a certain region, it's more likely to become that region. So what can we do now with this? Fabian showed beautifully the methods that you can use to now map the organoid atlas to the primary atlas. Here is the primary atlas. This is, again, from Stan Linnerson's lab that was just published, so we really could only do this now, since this came out. Before, there wasn't a good reference. And yeah, so now we integrated these. And when you now have them in the same embedding, then what you can do is you can ask for any of the cell types, how are they different? Is there any differential expression between them? So what I'm showing here is now differentially expressed genes between any of these neuron subtypes. And actually there was a lot of differentially expressed genes that were common between many of the subtypes. And so if we zoomed into those and did a geo-enrichment analysis, then we could see that there were quite some genes downregulated in organoids overall, actually across brain regions. And these were associated with synapse organization, neuron cell-cell adhesion, nervous system development, regulation of dendrite morphogenesis, so linking a bit towards maybe maturation. And of course, one has to consider that the ages are also not perfectly matching. But so generally, the genes that are downregulated in organoids are a bit related to neuron maturation, maybe, whereas the upregulated ones in organoid, they were generally enriched for canonical glycolysis, mitochondrial ATP synthesis, coupled electron transport. So there seemed to be this metabolic signature that was upregulated in organoids. This is not fully new to the field. We know there can be this cell stress in organoids. There were papers about that. The papers mainly focused on cortical organoids. It's actually nice to, or maybe not nice, but it's good to see this is actually across all the regions. So this is not specific to cortex. We see this across the neuron types. What we can now do with this organoid is we can ask, is there any feature of the protocols that were used for making these organoids that maybe lead to less or more cell stress? And this is what we did here. So here's the glycolysis score, which we took as a measure for cell stress. The primary cells, so they are very low on this. Now the organoid data sets that are ranked. And you can see here the ones that have the lowest cell stress. And actually now here are the different features that I hope you can see that. Guided, matrigel, so they can be guided, unguided, matrigel, yes or no. Matrigel in media, yes or no, so not full embedding in the solid form, let's say, but having it just in suspension. Sliced organoids that are further cultured, or are they shaken or spinned on an orbital shaker or not? And actually, we can see that there is an enrichment of having sliced organoids. There's not that many protocols, but they are really enriched for having low metabolic stress. And also, shaking them seems to also help, at least a bit. And so now we can really try to say, okay, this is maybe a feature of a protocol that we should use in the future for upcoming protocol developments. Something that was interesting is if we now compare the transcriptomic similarity overall, we don't just look at the differential expression. We just say, how similar is my neuron subtype A in the organoid and my neuron subtype A in the primary brain. Then you can get this heat map where now we have all the organoid data sets and all the primary ones. And generally, you can see the unguided protocols, well, they make usually in a given organoid is different regions present. So you can see kind of red for similarity that are across all the different cell types here. These are the different regions, telencephalon, then we dorsal ventral, and then we have diencephalon, we have midbrain and hindbrain. And so we see them all present, and the similarity is actually quite high across. When we now guide the organoid towards cortex, let's say, then you see high similarity to cortical cells, and you just have less other cells there. That's why there's a lot of gray here. But actually, we find that oftentimes there are off-target cells. So you aimed for cortex, but you also get some ventral telencephalon, and actually sometimes you get some diencephalic midbrain and hindbrain cells. It was quite striking that if in this guided organoid, you guide it for cortex, and you get other cells, they seemed less similar to the primary. So maybe because you provided a morphogen that makes the tissue become cortex, and now still some cell developed that was a midbrain cell, it is somehow a bit perturbed because it saw the molecules, the cues for cortex. So this was quite interesting. But yeah, generally the similarity is quite good for any of them. So if we guide towards ventral telencephalon, we see high similarity to ventral. If we guide to the thalamus, we see high similarity to thalamus and so on. So now what we can also do is here, we can, as I said, we bring the primary and the organoid atlas together. What you can see here now is that we calculate how present is any is any of the primary cell types in any of the organoids. So if you see blue here, then you can see that we find a lot of organoid cells that have this same cell type as is present in the primary brain. But there are also these red regions, which are regions where we just simply have quite a depletion of these cell types in organoids overall across all the protocols. And these are now very interesting to look at. So what jumps out directly is these non-neural cell types. So the cell classes here are vascular, immune, so microglia, endothelial cells are missing, which makes a lot of sense. Also some other erythrocyte and so on. These are fully missing or super depleted. If we look at the neuronal ones, we can highlight here in red some strongly underrepresented cell populations, neuron populations in thalamus, midbrain, and hindbrain. That would be very interesting actually now to follow up on. We actually would want to make more of those because there might be diseases affecting those, and we should come up with protocols to make them. So how to systematically establish protocols to generate these underrepresented cell types? I will start with these non-neural ones. Well, this was our 4Inter project, actually, to make microglia, and this has been in collaboration with Beatrice Vinno's lab, to make microglia in vitro and actually bring them into the organoids. And Simon Schäfer is here. He is also an expert on this topic. Where is he? There, yes. He's also an expert on this topic, working in Rusty Gage's lab on exactly that as well. And taking it further with transplantations. But yeah, so we then used protocols to differentiate microglia-like cells in vitro, and we can co-culture them, and they actually infiltrate the brain organoid. And you can see very nice tiling of microglia in these organoids. And I want to just mention quickly that we sequenced the control microglial cells that grow in 2D culture. We also sequenced the co-cultured microglia cells after bringing them in, taking them out again, and sequencing them. You can see in this UMAP that there are kind of two leaves, and the control and the co-cultured microglia are really on the opposite leaves. So they mature and differentiate on kind of different trajectories, even though they all express IBA1 and different markers for microglia. And what we then did is actually we would take the control and the co-cultured microglia and compare them to a primary reference atlas. Again, bringing the reference in, bringing them together and comparing them. And we could actually see that in the co-culture, the microglia become much more similar to the primary microglia than on this 2D culture, which makes a lot of sense because these are cells that constantly survey their environment. And so this 3D environment really helps them to become more microglia-like. There's still a lot to be done because they are not 100% like the microglia, and some cues are still missing, and we are further working on this. The other aspect is vasculature, and I'm just like quickly highlighting that there are different approaches. I think a lot of groups work on that. We for example, also work with these blast vessel organoids that are really powerful, and you generate not just the endothelial cells, but also the supporting pericytes and smooth muscle cells in one organoid tissue, and a goal could be to co-culture them with brain organoids. Another goal is to use these, or another approach is to use engineered human endothelial cells and bring them into the brain organoids, which also works where you can, for example, induce ETV2 as a master regulator of endothelial cell fate, and potentially then, even perfuse this on microfluidic chips. This is all work in progress, so I'm not going to talk more about that.